we're back. Today is going to be our last class, but I'll always be around to answer questions for you if you need them questions answered. Just let me know. Yeah. And today we are going to go over the genetic engineering process with human cells. So we should start shipping the cells that people order tomorrow. So you should hopefully get them by Friday, the people who needed more cells. So expect them around Friday. So after you've done the antibiotic selection, you should have found a concentration at which the G418 antibiotic kills about 99% of all the cells. If you're having trouble figuring out what that actually is, just take pictures of your wells and post them, and I can try to help figure that out. But take pictures and post them, and I'll try to help you determine what 99-ish% is. All this stuff, it just comes with experience. It's kind of intuition and stuff like that, right? So don't expect to like have to get it on your first try, because that's not the case with these things. These things they take time to be able to intuit, to be able to just like recognize, oh, like that's what all the dead cells look like. Even me, it takes time for everybody. So now what you should do next after you figure out the antibiotic concentration that works best for your cells is you should start up a new plate of cells. Or if you don't want to start up a whole plate, you could start up 4 or 8 wells or so. 4 wells will probably be sufficient for your first experiment. Now, when you're doing an experiment like this for the first time, don't go all in on the first, experiment. Only do 2 trials to see if it works. If it doesn't work, then you can learn from your mistakes and go back and figure it out. So the first thing that you're going to want to do before you start doing anything, whenever you're trying to do something with your cells, genetic engineering, antibiotic testing, all stuff like that, the first thing you want to do is change the media. Changing the media, make sure that the cells are in a good, healthy, stress-free environment, provides increases the probabilities that your experiment will work. So the first thing that I do is just take your plate and change the media and stick it back at 37°C and let it sit in the new media for like an hour or 2 if you can. Let it get used to the media, let things, let the cells settle down, get used to it, the condition, the environment, and stuff like that. Not be stressed out. After that, you can begin your experiment. Again, always make sure your media is as close to 37°C as possible, at least room temperature. And make sure you sterile filter it, right? The number one problem with contamination, twofold. Cleaning your environment, right? Get some Lysol wipes. These things are like, $5 from Amazon. Clean your environment. Get some Lysol wipes. Back when I first started, I would contaminate everything also, so don't worry. But then I just started being, very religious about cleaning and understanding what you can and can't do. Clean your skin a little bit, get all that dirt and bacteria off. Make sure you don't transfer that stuff into your cells. Again, it just comes with intuition and stuff like that. And also, don't try to be next to like an open window or a blowing fan or shit like that, right? It's just like, you don't want stuff blowing in the direction of your cell culture. You don't want open air coming on it when you're working. So, try to be in an environment that has little airflow and stuff like that. Kitchen table, if it's in the middle of the room, perfect, right? So, we're going to assume today that I already replaced the media on my cells, right? So, before you replace the media on your cells, make sure you look at your cells and everything like that. Make sure they look healthy and good before you do the experiment, right? And healthy, it's tough. It can mean a variety of things. I've seen pictures that you all have posted, and I've seen a ton of pictures that I have, from cells that I have. And, it's a difficult thing to pinpoint exactly what a healthy cell will look like. So try not to stress out too much of it. If your cells are adherent, you can probably assume that they're doing kind of okay. So if your cells are sticking, you can assume they're doing kind of okay. If they're not even sticking, or if they look super tiny and, not good, so one of the things I've been trying to do lately is figure out how to get these cells to last longer at room temperature or in the fridge. So I've been, letting them grow not at 37C. And these cells, they look pretty rough. They don't adhere really well. They're very sparse, right? So you see like some here, you usually see them individual, right? You don't see them packed together in like a colony clone. That's what HEK293 cells are well known for, is like forming these, like cells that like to group together and form these like, packed together colony type things. So if you're seeing those and you're seeing the cells adherent, just go with it. Do your best. That's all you can do, right? The most likely things, right, are always just temperature and media. So try to keep your temperature as close to 37 as you can. Getting one of those infrared thermometers is great, because then you can measure temperature. Having something that is a stable temperature environment, like a cooler, and sticking your heat mat in the cooler, like I said last week, works great. It helps keep those temperature fluctuations down. Also, putting something between the heat mat, I never really mentioned this. I didn't think it was that important, but the more I think about it, like I always put something between the heat mat and my culture, between the heat mat and the thing I'm incubating. This provides a little bit more barrier to the temperature fluctuations, because the heat mat can sometimes get hotter or hot, and it helps keep it a little more, in my experience, a little better, a little more stable, the temperature. So maybe give that a try, especially with the condensation. If you're getting that large temperature differential between the bottom and the top of the flask or the bottom of the top of the plate, that's when you're going to start to see that condensation. So, try to decrease that temperature differential if you can. Cooler works well. Give that a shot. All right. So, we changed the media on our plate. So the the next thing that we're going to want to do is we're going to want to mix up our components. So there's three things that are required for the transfection experiment, the one that we're doing at least. The first is the transfection reagent, Calvecten. Now, Calvecten is a general purpose transfection reagent that uses polymers to get DNA into cells. Now, Calvecten is pretty cytotoxic. Now, what cytotoxic means is that it's going to kill cells. So, like most transfection reagents with mammalian cells, they're going to kill the cells. So, you don't want to leave them in there forever. Generally, you want to leave them in there for maybe about 18 hours, 18 to 24 hours at the max. And then you want to change the media. And you'll see this when you do the experiment. When you do the experiment and you add the Calvecten, you'll see a lot of cells will probably die. It just happens. So try to keep the transfection reagent in there for a minimal amount of time. Make sure that you can take it out after about 18 to 24 hours. If you're going to have to leave it there longer because you have other commitments, obligations, don't, because your cells are going to be all dead. So you have to keep the timing on this pretty good, and go for the lower end. Don't go for the higher end on this one. Because if you have a lot of cells if all your cells are dead before you even start the antibiotic selection, it's not going to work out too well. So what we have is we have a plasmid. It's a mammalian plasmid. I don't know if I provided the plasmid map. I will do that. Make sure somebody reminds me if I forget. The plasmid is a human follistatin plasmid. So it's CMV, follistatin 344. So it overproduces the follistatin protein, right? Follistatin traditionally is a myostatin inhibitor, and when you inhibit myostatin, you see increased muscle growth. Now, obviously, these are just cells. They're not muscles. So you're not going to see any increased muscle growth in these. But the plasmid also has a selection cassette. The selection cassette is for neomycin or G418. Jet Optimus comparably cytotoxic. Honestly, Kent, I don't know. I've never used Jet Optimus before. It's something you can test yourself, right? Most all transfection reagents are pretty cytotoxic, so just you test it yourself. What you can do, Kent, is you can grow up a plate of cells and just do like I think for Calvecten it's like 3 microliters per 1 microgram of DNA. So I don't know what it is for Jet Optimus, but you can see how much it is for like 1 microgram of DNA and add it to the well and just see what happens. Do a control, do a Calvecten. If you need more Calvecten, again, hopefully we'll list it on the website soon. A lot of crazy stuff is going on, obviously, here. So we're really busy, and it's madness. Everything is emergency at all times. But we'll try to get some Calvecten and other stuff, more plasmid and everything, so you can try the experiment over and over again if you need to. But a lot of this stuff, there's not really scientific literature out there comparing like different transfection reagents and their cytotoxicity. maybe you could find something. If you do, let us know. But otherwise, it's something you just got to test yourself, basically. So, but it's also just like whatever works, right? If something works for you, keep with it. Keep doing it. That's the most important thing, is that it works. Calvecten is pretty inexpensive reagent, and the company that sells it is really great. Jewel, what media? What media are you talking about? Recipe for the media. So Calvecten is pretty great. So, I go with it. Inexpensive. They ship, I think, they ship to me, so I imagine they'll ship to anybody. So go for it. Try it out. See what's going on with Calvecten. The recipe for L15 media. You can search that online, I'm sure, Jewel, but most people buy it as a liquid. We bought some as a powder, which I didn't even know they sell. We're going to test it out and maybe list it on the website. But that's it. Like L15, you're going to have to search for I'm sure you could find it anywhere online if you just type in L15 recipe or Leibovitz media recipe. If you're talking about how much FBS and antibiotics to add, I think it's on the label. It should be 10% serum, 10% FBS. You can also use different other serums that are cheaper. You can try out NCS if you don't want to break the bank or other stuff. The antibiotics, 100 micrograms AMP, 100 micrograms strep, and 50 micrograms gent, I think we use. Other people use different. Some people just use penicillin and streptomycin, but again, go with what works for you, and that's what we're doing. So you got your Calvecten, you got your DNA, and then you got your DMEM. The only reason we use DMEM here is because the protocol for the Calvecten says to use it, and it works. So I don't like to mess with it. You could probably get away with using L15, but just follow the protocol that the company says. Don't fuck with shit if you don't have to, and do it. So the first step that you're going to do is you're going to take your Calvecten, and it's going to be, there's only a tiny amount in here. I think we sent like 15 microliters. But 15 microliters should be enough for like 5 different transfections. So it should be plenty. So I think you need about 3 microliters per transfection. So, it should be enough for 5. And again, we will put it up on our website soon so you can purchase more. So there's going to be a tiny amount. Make sure you flick it down, right? If you don't see really any in there, flick it with your wrist like that, so that it all settles on the bottom. DMEM and plasmids. So you're going to need 50 microliters of DMEM. Now, what you're going to do is you're going to mix the Calvecten, DNA, and DMEM. And the Calvecten is going to hopefully form micelles, like little mini cells, basically, around the DNA to shield it, to help it get through the membrane of the cells, right? So the protocol is, what you're going to do is you're going to mix the Calvecten and DNA. 1 microgram of DNA is 10 microliters. It's 100 nanograms per microliter concentration. And you're going to let it sit for a while, about 10 minutes. Try not to let it sit too long. The stuff with mammalian cell culture is a little more, it's a little more finicky, well, a lot more finicky than bacteria and yeast. So, with bacteria, if it's like do something for an hour, or yeast or something like that, right? You could do it for a half hour or 2 hours and it doesn't matter. With mammalian cells, it's not like that. Like, you got to be a little bit more precise on your timings and your temperatures and stuff like that. So when it says something like 10 minutes or something like that, do 10 minutes, okay? Now, what you're going to want to do is you're going to want to do one sample with DMEM and plasmid and Calvecten, one sample with DMEM and just Calvecten, and one well with nothing, right? So the Calvecten is going to affect the cells by itself. So you want to be able to see the effect that just Calvecten has. Now, what will also happen, what I've seen, is if you try to use like a really bad DNA prep, it can also kill off a lot of the cells, right? So if you have a DNA prep that is really shitty and has a lot of contamination and stuff in it, it will also kill off the cells. So it's good practice to do the Calvecten and do the Calvecten DNA separately, just so you can compare the difference between the amount of cells that are dying. Hopefully not too many cells die. This is also, you probably want to make sure that your confluency is pretty high when you're doing this experiment. So 70% or higher. You want to have a lot of cells in there because you're going to lose some cells in the process. The same thing happens with other transfections. So when you're doing bacterial transfection or yeast transfection or stuff like that, what's going to happen is a lot of the cells are going to die in the process. But the thing is with bacterial and yeast cells, you're not looking at it under the microscope, so you don't really notice it. But with the mammalian cells, you're looking at it under the microscope, and you're going to notice a lot of the cells dying. That's okay, right? So after you add that's the Calvecten and DNA and DMEM to your well, you're going to want to let it sit, like I said, for 18 hours. And then what you can do, if you want, is rinse out the well with PBS to remove any dead cells that are hanging out. Not that the dead cells really matter too much. I just, I like to keep my plate wells clean, free of all the contamination from dead cells and other junk. So if you want, rinse it out with PBS, remove all the dead cells that are hanging out, and then add in fresh L15 with, the media that you have with the antibiotics and serum and everything like that. And then you're going to add the G418. Now, you did the experiment hopefully last week, or you will do it when you get your cells, and you're going to find the concentration at which G418 works. Generally, that's going to be somewhere between 400 micrograms per ml to 800 micrograms per ml. It could be higher, but those are generally the numbers I've seen. So if it's like really low, if it's like 200 micrograms per ml, something might be wrong. So just give me a shout, take some pictures, so we can make sure that everything is correct. And if it's really high, if it's like 1200, 1600, 1800, something like that, also give me a shout out so we can try to figure out what's going on. It might just be normal for your cells in the environment that you have them in, but it could also be, something that you missed. Now, once you start selection, you also have to be pretty rigorous about it. So every 2 days, you're going to want to change the media, give the cells fresh media and fresh antibiotic. So every 2 days, 2 to 3 days, but do your best, right? You're trying to keep the cells as healthy as you can by giving them, a healthy living environment as much as you can. So switching out the media helps with that a lot, right? Giving them fresh media. Make sure that you add the G418 still, right? You want to apply that selection pressure. Now, what you're waiting for is what they call the The massive cell die-off. So what you're going to see is, maybe after like 3 or 4 days, something like that, you're just going to see a bunch of the cells start to die. Same with like your antibiotic selection plate. So you're going to see a bunch of the cells start to die. And that's when things are working. What you should look for is you should look and see how many cells are surviving, or if there are surviving cells. If you have like very little surviving cells at all, something probably went wrong with your transfection. So if you killed off like 99% of your cells, it's generally not a good sign. But you never know. You could have some living, growing cells there. But yeah, you want to keep track of how many cells are surviving during this whole process. And you should, after, like I said, like 3 or 4 days, have massive cell die-off. And you want to continue to add the antibiotics until, like 7 days or so. After 7 days, what should happen is there should be some cells that are growing, some colonies that are formed, right? You know how HEK293 tries to form those little clumpy colonies? You should see some of that. And that's when you know what's called, you've created a stable cell line. Now, a stable cell line is just a cell line that has taken up a plasmid and hold on to it with the application of selection pressure. So now you have a HEK293 that overexpresses follistatin and is neomycin G418 resistant. The important part is not those things, it's that you learned how to transfect these cells, right? Because now you can do it with anything. Now you can transfect other plasmids that you want to see what happens to the cell, see what it does. What I'm trying to do right now is, figure out protocols to where you can purify your own primary cells with, from your own tissue or from animal tissue that you purchase, so that you can start doing primary cell culture, engineer cells to do interesting and cool things and actually see the results, right? Because HEK293 cells are kind of just like general purpose cells to mess around with, to do scientific experiments with and figure stuff out. But if you really want to start doing more cool stuff, you need to start using like specific cell lines, right? Like, doing experiments on muscle cells, or doing experiments on neurons, and doing experiments on heart cells. Now, you're probably not going to want to get your own neurons or heart cells, but you can purchase these from companies for not too much money, a few $100s. So it's something that's within the realm of possibility. Once you start understanding how to do cell growth and cultivation and how to transfect them, you can work with lots of different cells. I wouldn't recommend tomorrow you go out and buy, heart cells or muscle cells or neuron cells, because they're probably going to require special media and be difficult to cultivate. First, I would say just get comfortable with HEK293 cells. Mess around with them, do a bunch of experiments on them, test different temperatures, test a lot of different things. Get really comfortable with them before you move on. Otherwise, you're probably just going to waste a lot of money if you just try to wing it. People who just try to do like neuron cell culture and don't have much experience with cell culture, a lot of times they just end up messing it up and they blow their money, right? So like, get comfortable with cell culture, get comfortable with cells, it should really help. Jewel, does it matter if you get vented or plug caps for tissue culture flasks? It really doesn't. There's no, since we're not doing, since the type of cell culture you're doing in this class or with L15 media doesn't require CO2 gas exchange, sealed caps are just fine. You don't need vented caps because the gas exchange isn't really taking place. And the oxygen inside the flask and CO2 and everything like that is plenty for the cells. So, plug caps or sealed caps or whatever they're called are probably what's best. Just like one less thing to worry about, making a mess with, in my experience. So I like the plug caps, sealed caps. They work well for me. All right. So if you've gotten up to this point, hopefully this last experiment, it's gonna take a little, it might take a try or two, and that's okay. Again, like, don't be discouraged if you can't do these things on the first try. I had trouble doing everything on the first try, right? Probably failed so many times. It took me a while to get all this stuff working, so don't feel discouraged if it also takes you a little bit. But just follow the protocol, go through it, ask any questions if you have questions, and hopefully it winds up good at the end. Anybody have any questions about the class, about the transfection, about anything going on with the class? Any questions? Any questions? Again, make sure you get in your media orders and order some cells if you need them. We're going to try to get those out tomorrow. This is going to be the last class. We've taken you to genetic engineering. Like I said, I'm working on some stuff right now regarding primary cell culture, so hopefully we can do something with that in the future. But I think this is a good start, getting it to genetic engineering in this class. Cell culture and engineering. I mean, gosh, this took me, 6 months longer to figure out all this stuff, and you guys are learning it in just a few weeks. It's pretty crazy. So any questions? Any questions about the class? Any questions that anybody has? Nobody really has any questions. Post your questions in Google Classroom. I'm doing my best to try to answer them and get to you as quick as possible. If you need something quick, it's probably better to email me or something like that. No, it doesn't really make sense to wash the trypsin off of cells. No, this is not a plasmid from David Ishii. Oh, nice. Nice tissue culture incubator. I'm jealous. I don't even have a nice tissue culture incubator. Maybe one day. Yeah, Jewel, I think it's probably best to put the flasks and plates on stuff. These heat mats, sometimes the temperature regulation can be a little fluctuating. So give it a little barrier. Nice. $15. I wish I could find a cabinet that cheap. No, it's actually good. What we use here is we use chicken egg incubators. The reason we didn't use them in the class is they're pretty big, and so the shipping cost would have just been too much to make it reasonable. We were trying to keep the cost of the class pretty low. So the chicken egg incubators probably would increase the cost of shipping a bunch. So we didn't add them. But if you want, you can go on like Amazon or eBay and find chicken egg incubators and just take out the chicken egg part and just keep the incubator. They are moderately good incubators. They're not the best. I mean, when you're paying for something that cheap, right, it's not going to be amazing. But they work pretty well, and they keep the temperature inside pretty well regulated. That's perfect, Heat mat fits right in there. But yeah, just keep it going. Do some cool experiments. One good place, I know Kent had trouble with GenScript, but maybe you got lucky with them. They don't seem to bother me. Just make sure you pretend like you're from a company and you have a PhD. I even use my Gmail address. That's why I'm surprised. Kent had trouble ordering from GenScript. But GenScript has this thing called the ORF database. ORF stands for Open Reading Frame, which generally just means gene. And you could basically buy any gene on a human plasmid for like $100. So if you want to test out the effect of a certain gene on these HEK293 cells, you can do it. And you can even get them GFP tagged and other cool stuff. So it's a really cool thing that I like to use. That's where the follistatin is from. It's from GenScript. Well, maybe I shouldn't have said that. Hopefully GenScript isn't listening. But yeah, it's from GenScript. So you should check them out. They have, I think, pretty much every human gene in there. So, try something fun and new. Let us know the results. You all are the first group of people in the wild doing human cell culture. All right. If there's really no more questions, then like I said, if you have questions, post in the Google Classroom, post in the Facebook group, email me if it's urgent. But let's get this stuff working. I want to see some people with some successful transfections. We'll send out the cells and media tomorrow to people who ordered more. So you can start doing that up again. If you need more cells and media, look in the Google Classroom. There's some links where you can purchase them. Otherwise, yeah. Thanks for joining this class. Hopefully next year we'll have some new cool stuff to do with these cells. Hopefully some primary cell culture stuff, or even CRISPR. I've also been working on CRISPR with these human cells, so that might be fun also. All right. I will talk to you later. Have a good one.